All right, let's dive into a really fascinating story about how a simple statistical error can turn our whole understanding of cancer survival completely upside down. It's a true data detective mystery, and the stakes couldn't be higher. So it all kicked off with this question. A question that seems, well, impossible, right? Researchers were digging through a huge cancer data set when they found something that just didn't add up. It was a genuine medical paradox that pretty much went against everything doctors thought they knew. Okay, so this is the graph that started it all. See that pink line way up at the top? That represents the Basal-like subtype, which is known to be super aggressive. But here, it looks like it has the best long-term survival of any group. For any clinician, this was a massive red flag. It just flew in the face of decades of clinical experience. And look, this wasn't some small, obscure study. This finding came from the Cancer Genome Atlas, the TCGA, which is a massive and highly respected database. This shocking result meant they had to ask a serious question. Was all the established biology wrong, or was something sneaky hiding in the data itself? Yeah, the raw numbers were telling a really strange story. A median survival of 7,455 days. That's over 20 years. For one of the deadliest forms of breast cancer, it was totally unexpected. So the scientists had to become data detectives and really start investigating the data itself. The researchers had a hunch. They suspected the problem wasn't actually biological. Instead, they thought there might be a hidden flaw, a glitch in how the data was collected and organized over time. And they quickly honed in on a potential culprit called follow-up time bias. The best way to think about it is like a race, where different runners have totally different finish lines. If one group gets to run for much longer, you can't really compare them fairly to the groups that had to stop way earlier, can you? And this table just lays it all out perfectly. You can see that patients with the Basal-like cancer were followed for a maximum of over 8,600 days. Now compare that to the normal-like group, only tracked for about 4,200 days. The Basal-like group had more than double the observation time. That creates a huge, unfair advantage in the survival stats. So to solve this mystery and get to the truth, the researchers had to find a way to make the comparison fair. They needed to level the playing field for every single patient group in that dataset. And their solution came down to this one number, 4,267 days. Why that number? Because it was the shortest maximum follow-up time out of all the groups. This number was about to become the new universal finish line for everybody in the study. The method they used is called data truncation, and it's actually pretty clever but simple. First, they identified that common endpoint, that 4,267-day mark. Then, they basically just digitally cut off any data for any patient beyond that point. And finally, they reran the entire analysis on this new, fair, and totally leveled dataset. So, with the data all cleaned up and the playing field finally level, it was time for the big reveal. What did the true survival rankings look like once that bias was gone? The difference is, well, it's absolutely staggering. On the left, you see that flawed data from before, with the pink Basal-like line floating on top. But now look at the image on the right. After the correction, the whole picture of survival changes dramatically, and you can see all the lines now end at the same exact point in time. And the rankings just got completely reshuffled. Look what happened. Basal-like, which started in first place, plummets all the way down to fourth. And the HER2-enriched type, another aggressive cancer, drops from second place to dead last. Suddenly, the corrected rankings lined up perfectly with what doctors have known for decades. The data was no longer lying. So what's the big takeaway from this whole data detective story? It's about so much more than just a quirky statistical error. This whole investigation teaches us such a powerful lesson. It shows that data can create these very convincing illusions that go against reality. And it proves that the methods we use to analyze data are every bit as important as the data itself. It's a critical reminder that questioning weird results isn't a failure. It's a vital part of science, especially when patient health is on the line. And I think the researchers' own words sum it all up perfectly. Getting the methods right isn't just some boring technical detail. It is absolutely essential if we want to turn these giant datasets into real, life-saving insights. It really makes you wonder, what other illusions might be hiding in our data, just waiting to be found?